Here's a single nucleotide. It has a sugar, it has a base, and it has a phosphate. Can you tell, is this deoxyribose or ribose? We're missing the oxygen here, so this is obviously deoxyribose. Deoxy, because you're missing that oxygen. If you notice these numbers, what we do is we label the carbon atoms from 1 to 5, going clockwise from the oxygen. So this is carbon 1, carbon 2, carbon 3, carbon 4, and this is carbon 5. So going clockwise around the ring, that's how we label them. And this is going to be important because a nucleotide has a direction. We call it 3' and 5'. And so the 5' region is up here, the 3' region is in here, and to cut to the chase, all nucleotides are always added in a 5' to 3' direction. In other words, a new nucleotide would be added at the 3' side down here. When you start adding nucleotides together, we end up with a chain of nucleotides. Here's a single chain. Here's a single nucleotide. Here's your nucleotide. This is what it looks like, the phosphate, the base, and the sugar. And they are joined by bonds. And there's a bond right there. This bond right here between the ribose, or deoxyribose of the previous molecule and the phosphate of the next molecule, and that is called a phosphodiester bond. And it's joining the 3' oxygen and the 3' carbon to the phosphorus of the next molecule. So that'll be down here. So this would be the phosphodiester bond right in there. There were many people contributing to the discovery of DNA, and a gentleman called Edwin Chargaff is one person, and he was looking at the amount of A, C's, G's and T's in any given DNA sample. And here's some of his original data. These are different organisms down here. Phi-X174 is a virus. E. coli down the bottom, as you know, is a bacteria. And here is some data, the percentage of A's, G's, C's and T's. Here's the AT ratio, the GC ratio, percentages of G's and C's, percentages of A's and T's. What you want to see is that the amount of A's and T's are generally very similar, and the amount of C's and G's are very similar. Let's go to humans here. In humans we see 29% A. If we go over to T, we see that it's about 30% T. If we go to G, 20% G and 20% C. So it's same numbers of C's and same amounts of same numbers of G's and C's and same amounts of A's and T's. Ditto. Let's go to yeast down here. It's about 31% of A's, 33% of T's, just about 18 and 17 there. So a little bit different. This one up here is interesting. 24% A, 31% T, 23% G, and 21% C. So it doesn't seem to follow the rules. And I'm going to ask you in class why you think that might be. Anyway, what we get from here, what Chargaff deduced was that there are A's and T's and they pair together. And there are C's and G's, or cytosine and guanines, and they pair together. And here's an example. Here's an adenine, and it's pairing with a thymine. And in between, there are hydrogen-bonded bases. So this is a hydrogen bond right here. And from your chemistry, you should know the properties of hydrogen bonds. And again, I'll ask you that in class as well. Watson and Crick then proposed that the DNA has these two nucleotide strands. They're called polynucleotides because it's multiple nucleotides. And they're in opposite directions. And we've drawn two lines here, and these are the two strands of DNA. And when we draw a line like this, this is the 5' end, and this is the 3' end. So this will be 5', and this will be 3'. And so these are anti-parallel. These are opposite strands. anti-parallel strands. And then these strands are, of course, are all wound up in a right-handed helix. Thus we have a double helix, right? 2 strands in a helix, double helix. So the model then is 2 polynucleotide chains, opposite directions or anti-parallel, coil up to form a helix. The sugars and the phosphates are on the outside. Here's the sugar, here's the phosphate, here's the sugar, here's the phosphate, and the bases are on the inside. Here's a T bonding with an A and a C bonding with a G. I'm going to ask you a question in class. Why are the bases on the inside and why are the phosphates on the outside? So some of the numbers involved in a double helix, we find that at the top of the screen here, the double helix is actually very, very small, 2 nanometers in diameter. The wavelength of blue light, for instance, is 400 nanometers, so it's quite small. Very little distance between each pair of bases, 0.3 nanometers, and there are 10 bases for each full twist of the double helix, thus 3.4 nanometers between each twist, basically. One of the interesting remarks made by Crick at the end of their paper announcing the structure of the double helix is that they mentioned that if you knew one side of the helix, you could predict the other side of the helix. And here's what we find here. If we know that one side of the helix is A, C, G, T, G, then we know that the other strand is going to be T, G, C, A, C. And we generally write this in a 5' to 3'. So here's the 5' area. So it's going to be 5', A, C, G, T, G, and the reverse sequence, the complementary sequence, is going to be 5', C, A, C, G, T, because of course this would be 3', and this is 3' over here, you see, this one right here. So how much DNA is there in a genome? Well, it really depends on the species. If we look on this slide, you see a bacterium such as E. coli has about 4.6 million nucleotides, 10 to the 6 there. And if we go down to humans, we find it's about 3 billion nucleotides. So there is a difference between organisms, between species, but it is not a linear increase. There are exceptions to this. But nonetheless, we have 3 billion nucleotides, and somehow we fit those number of nucleotides, which if you stretch them out, turn out to be 2 meters in length, 6 feet, 2 meters long of DNA in each one of your cells. How do we fit that much into a nucleus that's only 5 micrometers wide? We can take this double helix DNA and we take some proteins, and that will coil up the DNA into something that's much, much shorter. These proteins here are known as histones. These guys are here. These histones there are 5 different histones. There's histone 1, there's histone 2A, 2B, 3, and 4. And these last 4 are, there's 2 of each of those. And these 8 proteins all get together into a nucleosome. And that's what's coming along next. Here is a cartoon of a nucleosome. This is the histone core. This histone core contains those 4 different proteins, all times 2, so there's actually 8 of those proteins within the core. And histone H1 actually sits on the outside there. There is about 140 base pairs of DNA that wraps around the nucleosome core, and which leaves about 60 base pairs on the tails on the outside here. So a total of about 200 nucleotides per nucleosome. If we look at the bottom of this diagram, you'll see here's the nucleosome core with the DNA wrapped around it. Those nucleosomes wrap up, and they wrap up, and they wrap up, and they become what we call beads on a string. You see this here. This is beads on a string. That's actually a technical term, and it looks like this. This is the nucleosomes under an electron microscope. So these nucleosomes wrap up, and they wrap up, and once those coils wrap up, you become even more wrapped up and more coiled, and eventually it becomes a chromosome that we see under the light microscope. There's multiple layers of coiling that's going on. So every time our cells replicate, we have to replicate the DNA as well as all the other cellular machinery. And if we know the sequence on one strand of the DNA, it means we can replicate the other strand of DNA. It's known as semi-conservative replication. And so we take an original double helix, we split that original double helix into two single helices. You see how the original blue is separated. And then we create a new helix, that's the yellow here, on each of those original blues. And so each daughter strand and a daughter molecule, sorry, each one of these is a daughter molecule, consists of one original blue strand and one new yellow strand. Here it is in a little more cartoony. Here is the original DNA sequence. We can see we split the double helix, so there's now two single helices, and the new DNA is laid up against the old DNA and put together. Which way is DNA made? 5' to 3' or 3' to 5'? That's the question. There are a number of enzymes that put together DNA. If you're going to have an enzyme that makes a DNA polymer, then you have DNA polymerase. You simply add ASE on the end of the action that you want it to do. This replication can start in many areas. In a eukaryote, there are thousands of origins of replications. However, in a prokaryote, there's only one origin replication. Why do you think that is? There are also many other proteins involved in replicating DNA. There's to stabilize the DNA, to separate it, to bring in the new nucleotides, and so forth. Replication occurs at a replication fork. This is the point whereby those two old strands of DNA are separated and the two new strands of DNA are made. One is made continuously on one strand and one is discontinuous on the other strand, and this is to do with the 5' to 3' requirement for making new DNA. Here is a cartoon of a replication fork. This blue line is the old DNA, and the new yellow DNA you can see here depicted as arrows. This is the continuous line, this new DNA here. This is the continuous, if you can read that. And so this end is going to be the 5' end, and this little arrow is going to be the 3' end on this strand here. On each of the strands on the opposite strand, it's 5' here and 3' here. New DNA has to be made 5' to 3'. So the problem is, new DNA is being made here, and it's going away from the replication fork, which is up at the top. This is the replication fork right in there. So we have a problem, and Okazaki was the person who discovered this and came up with a theory as to what happens. You should probably watch a YouTube video. There are many YouTube videos on DNA replication. I suggest you go and watch a few. Here is a cartoon of what happens, and you should be able to draw and label a complete replication fork and understand the functions of each of the components. And we will go over this in class, of course. But more importantly, you should understand the functions and predict what will happen if one of the components is changed. That completes the chapter 8, and we will be asking questions in class, so study well.